lunch was awesome. And if you could finish what you're working on for lunch, it was fantastic. And so were the beverages from Alto Cinco, just 2 blocks away here in Syracuse. So if we want to grab our seats again, we'll continue with our presentation. We heard from North Carolina shortly before the lunch break, and now we're going to go to Tulane University for its presentation on bacterial persistence and the immune response to infection. So the lights are coming down, everybody's being seated, and I've seen Dr. Embers here. Hope she had a good lunch. She's from the Tulane National Primate Research Center. Ladies and gentlemen, from Tulane, Dr. Monica Embers. Doctor, it's yours. Thanks for being here.
All right, thank you. It's great to be here. It's nice and cool. So today I'm going to do something a little bit different, and I'm going to talk about the role that the immune response plays in infection, how it can affect both diagnosis and treatment. And so we've done a lot of research using the non-human primate model of Lyme disease. We've also worked with mice, and animal models are sort of our forte. But when it comes to persistence by the bacteria, this is the norm. This is not the exception. So in mice, they fail to clear the infection with Borrelia burgdorferi. And we know that the ability of the pathogen to survive through the enzootic cycle requires minimal host immunity. Now, we also know that primates appear to harbor a low-level persistent infection. So it looks a little bit different than it does in the reservoir host, which is the mouse and other species. And it appears also that there are immune evasion tactics that the spirochetes use to contribute to persistence in the incidental host as well. We don't know this as clearly as we do in the mouse model. So this was my first paper in the field. This was from 2004. And it's basically a review of all the things that Borrelia does to either evade or suppress the immune response. And you can see that there are both adaptive level immunity. There's interference at both the adaptive and the innate level, including the induction of anti-inflammatory cytokines, the tolerization of monocytes, sequestration of antibodies potentially in immune complexes, and phase and antigenic variations. So once, the immune system, the antibody response starts to recognize the Borrelia, they change their outer surface. Very clever. And this is what got me into the field too, is just all the things that Borrelia does in order to be able to persist. And so we have to think about immune suppression, not only from the spirochetes, but also from the ticks that transmit them. And so I'm not going to go through these data too closely, but basically on the left panel are macrophages, and on the right panel are dendritic cells from mice that were infected with Borrelia burgdorferi. And the white circles and squares are IL-10 deficient mice, and IL-10 is a suppressive cytokine. And what this lab found was that in the presence of Borrelia, both macrophages and dendritic cells increase their production of IL-10 and suppress the production of other cytokines that help to recruit the immune response to the infection. And in the IL-10 deficient mice, you really didn't see that happen at all. So what about the ticks? What we know is that there are factors in the tick saliva that do a lot of things. They suppress itch, so you don't feel them. They suppress the immune response. And in particular, when it comes to the adaptive immune response, we know that the tick saliva can suppress antigen-presenting cells and lymphocytes, both B and T lymphocytes. And so this is really important in the ability of the the host to generate solid immune responses to the pathogen. And when you think about vaccines and things like that, it's also something you need to overcome when you're developing like an anti-tick vaccine, which has come as an interest recently. So in terms of our research, we've done a good bit of work with both diagnosis and treatment. And I'm going to present to you a longitudinal assessment of antibody responses and how it varies in rhesus macaques. And also from the treatment side, an examination of the responses of individuals with clinical cure versus those who have post-treatment Lyme disease. So the clinical case definition of Lyme disease is either erythema migrans, which is considered pathognomonic, 5 centimeters in diameter, which is sort of absurd in my mind because that doesn't always fit the bill. And there was a recent study that just came out and it showed that the EM rashes in women were smaller than those in men. So we have to take sex differences into account. We have to take like skin color into account, all sorts of things. Or at least one late manifestation of musculoskeletal, nervous system, or cardiovascular involvement and laboratory confirmation. And the laboratory criteria involves at least one of the following: isolation of Borrelia burgdorferi, which rarely works. If it were in the blood and we could detect it, we'd have a direct detection diagnostic, and we don't. So it's only in the blood for a narrow window of time. And the demonstration of diagnostic levels of IgM or IgG to be burgdorferi, which is also highly variable. And I just did a breakdown of the original paper for the Western blot criteria and looked at it very carefully. And if you look, the criteria are based on Lyme arthritis patients. And Lyme arthritis patients have robust antibody responses. And when they looked at the neuroborreliosis patients, they didn't even meet the criteria for the Western blot, half of them. So it's really, and they set this specificity to 99%, which is really hard to do. So the Western blot is not good. It needs work. Diagnostics in general need work. And as I'm going to show you, the antibody titer between acute and convalescent is used in diagnosis, but this is highly variable and it depends on what antigens you use. Oops, go back. Okay, so in terms of the variation and suppression of B-cell responses, we have evidence in both primates and in mice. So we're gonna look at antibody responses after experimental infection with Borrelia burgdorferi. And to do that, I need to talk to you about a study that we did many years ago where we looked at how the antibody responses changed over time in rhesus macaques that were infected with Borrelia burgdorferi. And we used a number of different antigens. And what we found was this, and I don't have a pointer, but if you look, the lines that are red are the treated monkeys, and the lines that are blue are the untreated monkeys. And in the x-axis, you'll see the time course. And the y-axis is basically the level of antibodies over time. And you can see that for C6, which is a peptide that's often used in diagnosis, after antibiotic treatment in these 3 monkeys, you saw a significant decline. With anti-OSP-C, you saw a decline which was irrespective of treatment. For DBPA, another antigen, you saw high antibody titers throughout the course of infection. And then for another antigen, OPPA2, we saw a decline after treatment, but it was incomplete. So they're all different depending on what antigen you use. And then we published this and developed a test with 5 different antigens. We call it a multiplex test because you can test all the 5 antigens and quantify the antibody levels at the same time. And the way that we do this is to use a fluorescent bead-based assay. So we basically coat these in the green circle here, we coat these beads that are different colors with each antigen. And then we combine those, and then we add serum. And if there are antibodies in the serum that bind to those antigens, then we can detect them with the secondary antibody shown here with a little red star. And we can also quantify it because we have controls and we have standard curves. So this is another study that we performed in non-human primates, and we started with 10 animals, and we infected them by nymphal tick bite. So we used a natural route of infection. We waited 4 months, and then 5 of them were treated with 28 days of doxycycline, and 5 were untreated. And then we waited another 3 months, and we did some xenodiagnosis studies where we fed unfed ticks on the monkeys. And then finally, we did a necropsy and looked in their tissues for persistent infection. So, okay, I'm going to try to break this down for you because it's really important. So if you look at the light blue line, which is the antibody levels to C6 over time, in each of these monkeys, there's 5 untreated on the left and 5 treated on the right. You can see that for some monkeys, after treatment, the C6 titers decline, but for some, they don't. And you can also see for all of the treated animals that the C6 levels declined. But if you look at IK14 there on the top, we couldn't find any antibodies in this monkey. If you look at down here, IH11, the third one down, if you were to do an analysis at the last time point in the chronic phase, you would not see OSP-C or C6. And for the untreated, you would, if you did C6 and OSP-C, for IL-75, you wouldn't detect it. For C6 and INO5 at the bottom, you wouldn't detect it. So these are even untreated, and their antibody titers have declined over time. So I want to keep those 3 animals in mind: IK14, IH11, and IH22. Those are all treated animals. And these are the xenodiagnosis results. IH11, so basically after the ticks feed, the uninfected ticks feed on the animals that have been treated, then they take up a blood meal, and then we look inside the ticks for persistent spirochetes because they didn't have Borrelia before they fed. And it's a very sensitive means to detect persistent infections. So if you look at panel B, IH11, that animal was treated. Panel C, IK14, that was our seronegative animal that was treated. And we also used RT-PCR on the tick midguts, and RT-PCR detects RNA, and messenger RNA is produced from growing or actively dividing bacteria. So this basically tells us that the bacteria that are in the tick are still alive. And this is a immunofluorescent staining in the tissues of the monkeys. And with, we have 2 different antibodies. One is, one lights up with green and one lights up with red. But basically the top 3 are just controls. A, B, and C are just controls. D and E show you inflammation in the brain. F, that panel is from IH22. That was a treated monkey, and there are 6 different spirochetes in the brain parenchyma of this animal. The next panel is IH11, and then we can see green spirochetes in that animal as well. And we saw various levels of inflammation, mild to moderate inflammation in different tissues, and persistent spirochetes. I want to bring your attention to item C, panel C. That is IK14. That was our seronegative monkey. And that's a really beautiful spirochete in the heart tissue. So here we have examples of seronegative animals that we know are persistently infected. So let's move on to suppression of B cell responses in mice. And this was what I consider to be a seminal study by Nicole Baumgarth, who was at UC Davis and now runs the Lyme, a Lyme disease program at Hopkins. So she's one of the few people who's studied the suppression of immunity in animal models during Lyme disease. And what she did was this really clever experiment where she took a flu antigen, a flu vaccine antigen, and basically vaccinated the mice against flu, and had a group that was infected with Borrelia and a group that was not infected with Borrelia. And she looked at the number of antigen-specific antibody secreting cells after the infection. And what she found was, if you look at panels E and F, so the top, in panel E, you can see the top line, those are the mice that were not infected with Borrelia. And the bottom line, the low-level antibody responses, are mice that were infected with Borrelia. And then in panel F, the total antibody secreting cells from the bone marrow in the vaccinated mice and the Borrelia plus vaccinated mice. And you can see a significant difference. So basically, Borrelia infection is suppressing the immune response to this vaccine antigen. So we decided to do this in our animal model as well. And mice are really great models. They're also inbred, most that are used. Non-human primates are outbred, and they're a little bit more genetically diverse than we are, actually. So that's the bane and the benefit of this model. So everything that we usually see in humans with Lyme disease, we also see in the non-human primates. So we decided to test the hypothesis that infection with Borrelia burgdorferi non-specifically impaired B cell responses or antibody responses. So we had 9 male rhesus macaques that were enrolled, and they were split into 3 groups, infected only, immunized and infected, and immunized only. And primates are the best model for HIV. So we have a well-established HIV program, and we used an HIV-derived GP130 subunit antigen since we already knew what the response should look like in rhesus macaques. And then we infected them by tick bite. So this is what the experimental design looks like. The animals were infected by tick bite or not infected, then immunized and boosted twice at 1 month and 2 month intervals. And then 4 weeks later, we took blood and lymph nodes. We also collected throughout the process and looked for antibody levels to both Borrelia and the vaccine antigen. And we looked at B and T cell populations from whole blood and lymph node biopsies. And so this is the only piece of data that I'm going to show you. We obviously have a lot more, but with the knowledge that these are very outbred animals, there's a significant variation in their antibody response. But the one thing you'll see in this panel here, LD59, was the only animal that after 2 boosts did not generate any antibody responses to the vaccine. So that means that for 1 of 3 animals, the vaccine response was suppressed by Borrelia infection, which is not to be unexpected because they're very different, and not every human is suppressed. Not every human's antibody responses are suppressed in the same way. Then when we looked at Borrelia, antibody titers to Borrelia, we also found another animal circled here that was seronegative, was infected, And we were able to culture Borrelia from multiple tissues from this animal. So we know it was persistently infected and not detectable by our antibody detection assay. So what about humans? There are differences, where I'm going to show you some differences in antibody responses among early Lyme patients and in their response to treatment, and then some information about B cells in cured patients versus those who develop post-treatment Lyme disease. So post-treatment Lyme disease refers to patients who continue to have symptoms after antibiotic treatment is given. It can be caused by the induction of inflammatory responses. It can be caused by a continued active spirochetal infection, or it can be caused by irreversible sequelae from a previous active infection or autoimmune, which you're going to hear about later. And it doesn't mean that any one of these is acting on its own. It could be many of these factors, or all three of them. So I told you about the test that we developed with the 5 different antigens, and we decided to run that test with a panel of patients from the CDC and compare the sensitivity and specificity to the currently used 2-tier test and to the C6 test. And then we also looked for differences in PTLDS patients. And item number 2, this comes from a study out of John Alcott's lab at Johns Hopkins. And you can see that he characterized patients. He basically had a cohort of patients that all had physician-diagnosed erythema migrans, all received the same course of antibiotic treatment, and then he followed them for 2 years. And he ascertained them for clinical symptoms, functional decline, or neither. And so most of them were neither, shown in purple. Some had clinical symptoms only, some had functional decline only, and the 11% that had both, he considered to have post-treatment Lyme disease. So these are the CDC panels. I'm not going to go through this too intensively, but basically every patient that had early convalescent or Lyme arthritis or neurolyme was positive by our test. But I want to bring your attention to this this one right here, 112. That's a Lyme arthritis patient that we use to compare our next set of data to. And you can see that the Lyme arthritis patients, in our hands, they've always had the highest antibody responses. And this just shows basically how well our test compared to 2-tier testing and C6. I'm actually going to skip over this. Okay, so these are the patients from Hopkins. The post-treatment Lyme disease syndrome patients are in the bottom blue. The healthy ones are on the top, and the residual less severe cases are in the middle. So there's 10 of each. And I thought that the post-treatment Lyme disease patients were going to have lingering antibody responses. So they're going to have a lot of antibodies because they had a persistent infection. That wasn't the case. Instead, what we're finding is a trend wherein patients who go on to be cured with doxycycline have good antibody responses to begin with. And so if you look, 9 of the 10 patients that returned to health generated strong IgG, which is antibody, to multiple antigens on the Bioplex test. Whereas all of the patients characterized as having post-treatment Lyme disease either showed weak responses to all the antigens or produced a titer to OPPA2 that did not decline. And we think this might be a good antigen to test chronic patients. So if you look at it in a figure, this is what it looks like. So the left is the response to C6 in each patient. The middle is a response to OSPC in each patient, and The right is a response to DVPA in each patient. And you can just see that the post-treatment Lyme disease patients have very poor antibody responses compared to the healthy. Then there was a study that came out of Bill Robinson's lab, and basically what they took the same population from the Hopkins group, and they looked at what are called plasmablasts. So these are antibody secreting B cells. And for those that were untreated, if you look at the untreated stage, before treatment, the patients who went on to have persistent disease all started with low-level plasmablasts. So the ability to generate an antibody response is really important for good outcomes with antibiotic treatment. So what we found is that host antibody responses appear to be predictive of clinical cure, and just basically knowing what doxycycline does. It doesn't kill the pathogen. It's a microbiostatic antibiotic, so it relies on a good immune system in order to clear the infection. It basically stops the growth of the bacteria. And some of the explanations for weak responses, this is something I would love to delve deeper into, different human differences in antibody responses to infection. But it could be host differences in the ability to generate humoral responses, possibly via isotype switching. One of the weird things about Lyme disease is that patients can have persisting IgM, which is supposed to be the first antibodies that are produced, and then they isotype switch into IgG. So that could be failing in these patients. And strain differences among Borrelia and other co-infections could influence the host immunity as well. This is something that we really haven't studied, and it needs more attention. So in summary, the gold standard diagnostic tests detect antibodies to Borrelia burgdorferi, and these responses vary significantly among individuals and change over time. So it depends on where your snapshot is when you take that blood sample and how well the diagnostic test is going to work. Currently, the most recommended antibiotic treatment is microbiostatic, so it relies on a good immune response. We need more research, but several studies indicate that the humoral response to infection correlates with a good treatment outcome. And we know that Borrelia can manipulate the host antibody response, and this could depend on strain differences in host factors, which also is something that I think deserves further investigation. So with that, I would like to thank the members of my lab, my graduate students, my staff. They do all the hard work. I have a postdoc. We've received a great deal of funding from foundations, including the Stephen and Alexandra Cohen Foundation, the Bay Area Lyme Foundation, Global Lyme Alliance, and the Bill and Marion Cook Foundation paid for our non-human primate immune vaccination study. So with that, I'll take any questions.
Thank you, Dr. Embers. Questions, raise a hand. We'll get a microphone to your front row with a gray suit next to Brian.
Robert Lockhead, Medical College of Wisconsin. And first of all, I'd like to thank you for setting up my talk beautifully. Oh my goodness, this is great. My question is, I mean, you said exactly what I was thinking. Doxycycline is bacteriostatic. You still need a good immune system, antibodies. And, you know, little teaser in my study, you know, in about a third of patients that don't resolve their arthritis with doxycycline, if you give them ceftriaxone, which is a bactericidal antibiotic, not all of them, but some of them do seem to get better and clear the infection. So that relates to my question. In your study or in Bill's study, do you try IV ceftriaxone and see if that's more effective? I would imagine it would be, even though it's harder to, you know, give patients routinely, but
so
about that.
So for the humans, I don't know. This is all John Alcott's. I think he does follow up with ceftriaxone in those patients. We have done a great deal of work with combination therapy in mice. And what we found is that in general, monotherapy is ineffective at clearing the pathogen. But when we combine doxycycline and ceftriaxone, even sometimes with another antibiotic, we can get clearance. So, yeah,
That's good. That's valid insight.
Yeah.
Raise a hand. Other questions? Microphone coming your way.
Hey, Lauren Edline, SUNY Upstate. I have I won't take up too much of your time, but I have a little bit of a diagnostic case conundrum. Oh, sorry. I had a patient in my practice who was seeing neuro-ophthalmology and was admitted to the hospital for suspected meningitis, and they did an LP. And I had already suspected Lyme in her, and we had done all the diagnostics that we know to do, and they all came back positive, IgM and IgG, and was fitting with a lot of the neuro symptoms that she was having that they thought might have been meningitis at the ER. When they did her LP, the CSF came back negative for Lyme antigen, and she was told she did not have Lyme disease. So I wanted to know your thoughts on that.
Ooh, yeah, that should not be used as diagnostic criteria, and I don't think the IDSA even recommends that. So, yeah, you have to have an infection. You have to have infection in the brain specifically to see those antigens shed. And even in the brain, we don't find high numbers of spirochetes, whether the animal's been treated or not. So, yeah.
Raise your hand. Next quote right there.
Okay, so that was a lot of immunology stuff thrown at us. Some of us are not doctors.
Sorry.
It's okay. I think I got through it, but correct me if I'm wrong, please. My conclusion is the number one test that they're using to say, I have a bunch of symptoms and I'm going to go to my doctor and say, I think it could be Lyme disease. And they're going to give me a test, and the test is inconclusive. The symptoms are inconclusive because they vary so widely amongst patients. If they do decide that you may or may not have Lyme, and they go ahead and prescribe an antibiotic for you, that antibiotic is only going to work for you if you have a good immune system, which we don't know from the get-go.
Mm-hmm.
And whether you get the treatment or not, if you are one of those unlucky few who has a suppressed immune system, whether it's because of you personally or because the the bacteria is playing games with your immune system, if you are one of those unlucky people, whether you get the treatment or not is irrelevant. Whether they can detect it or not is irrelevant, but you're going to continue to have these mysterious plaguing symptoms for a very long time. Is that what you just said?
In a nutshell, if you don't have good antibody responses, you're not going to get diagnosed, and your treatment might not work. Yep.
Next question. Anybody? We got some time. Yeah. Speak right up.
Hi, Simone Seward from Upstate. So, given all of this information, how does this work for pregnant women and Lyme disease?
Ooh, that's another topic that for some reason has ended up to be controversial. If a pregnant woman acquires a known case of Lyme disease, amoxicillin treatment is generally very effective. If a pregnant woman or if a woman is infected persistently and becomes pregnant, we have no idea. It's a big black box, and it needs more research because there are a lot of cases of what would be considered perinatal Lyme disease. And the epidemiology just hasn't been done to support it. But the case reports have clearly shown that there can be birth defects and there can be transmission. I think it's not as prevalent as relapsing fever infection, because relapsing fever is in the blood. It's definitely crosses the placenta. And it's the most dangerous for the unborn fetus. if you acquire relapsing fever. What else has not gotten enough attention is Borrelia miyamotoi, because it's a relapsing fever spirochete. And if you probably won't get diagnosed because doctors don't necessarily know to run that test, and you will transfer it to your child, to your unborn child. So it's a big problem.
Who else for Dr. Embers? Yes, sir.
I think you showed. Okay, I think you showed another antigen OpA2. Were you saying that was a potential antigen to be used for correlating with the syndrome?
Yeah,
Okay.
we were hoping that it could be used as a antigen to indicate persistent infection. We just haven't had enough animals to show that, and there's no way to determine persistent infection in humans. But what we do know, and I didn't present this, is that if you look at post-treatment Lyme disease patients that were diagnosed at different points of infection, which is the SLICE 3 study at Hopkins, when we compared our test with C6 and 2-tier, we picked up at least 50% more Lyme disease in those chronic patients than 2-tier or C6. So I think even if it's not, it doesn't improve early diagnosis, it can definitely improve late diagnosis.
And questions? If not, how about a round of applause from Tulane for Dr. Monica Embers?